everyone, my name is Megan Bailey. I am an undergraduate researcher working in the Division of Animal and Nutritional Sciences at West Virginia University in Morgantown. Today's increased demands are forcing the livestock industry to require higher, more efficient animals. Due to there being an antagonistic correlation between reproductive performance, protein yield, and fat yield, the selective breeding that occurs to sustain these demands has a negative effect on reproductive performance. This negative effect presents itself as infertility among cattle herds. During early embryonic development, maternal transcripts that accumulate in the oocyte during oogenesis and folliculogenesis play important roles in early embryonic genome activation. Some maternal transcripts are oocyte-specific and known as maternal effect genes, which are needed for early development events after fertilization. Our lab aims to study novel oocyte-specific genes in cattle. Specifically, I'm working with a graduate student who is trying to characterize novel oocyte long non-coding RNAs, also known as lincRNAs. It has been documented that lincRNAs regulate a wide variety of processes. There have already been important lincRNA discoveries in human and mouse embryonic development. However, there is very little information on bovine oocyte-specific lincRNAs. The objective of this study was to characterize the expression profile for OOSNCR1 and OOSNCR5 in bovine tissues in order to validate previously published RNA-seq data. For my materials and methods, tissue samples were collected from local slaughterhouse, snap frozen in liquid nitrogen, and stored at a -80 degrees Celsius until analysis. Back at the lab, tissues were homogenized in trizol using a bead bug. RNA was isolated, DNAs treated, and finally, complementary DNA, or cDNA for short, was synthesized using a high-capacity cDNA reverse transcriptase kit. The cDNA was then put through reverse transcriptase polymerase chain reaction, or RT-PCR, in order to amplify both OOSNCR1 and 5 lincRNAs. The samples were put through gel electrophoresis with a 100 base pair ladder in order to visualize the gene. Previously, my graduate student had identified 1,536 lincRNA in bovine oocytes. Table 1 in my preliminary results presents the 6 predicted most abundant lincRNAs within the oocyte. OOSNCR1 and OOSNCR5 were predicted to be the most abundant, so they were chosen for further investigation. Moving into my results. The 3 gel pictures on the left side of my poster are the tissue expression profiles for RPL19, OOSNCR1, and OOSNCR5, using the tissues isolated from what I will refer to as the novel samples, meaning the samples that I collected this semester from the slaughterhouse and performed the RNA isolation on. At the top of the gel, you can see 14 different tissue types I collected and a negative control. Starting with RPL19. RPL19 was used as a housekeeping gene. It was expressed in each one of the tissue types at varying concentrations. OOSNCR1 was not detected in any of the tissue samples. OOSNCR5 was detected at very low concentrations in the fetal testis. So if you look down here towards the bottom gel, right here, there's a very faint line, and that is the fetal testis, or the gene being expressed in the fetal testis. Based on the results, we decided that something was going on with our data and started to troubleshoot. After asking multiple questions and performing RNA quality gels, we concluded that there was significant RNA degradation in our novel samples, yielding unreliable results. We decided to test our lincRNA using previously isolated samples that have been used by various students in the lab. I refer to these samples as proven samples, and there are 2 different fetal ovary samples. In proven samples, we saw a strong detection of RPL19 and OOSNCR5 in both samples. In OOSNCR1, it was detected at low concentrations in the fetal ovary in sample number 2. Using proven samples, we confirmed the novel samples failed and that OOSNCR1 and 5 are present in the oocyte. We were not able to confirm if they are oocyte specific because we need to run the entire tissue panel on known proven samples. So here we have the gel that was run with the fetal ovaries, and these are 2 RPL19 samples, and then here are the genes being expressed in both fetal ovary 1 and fetal ovary 2. In conclusion, RNA sequencing predicted that OOSNCR1 and 5 to be oocyte-specific, and the most abundantly expressed. The initial expression profile generated for OOSNCR1 failed to produce a band in the fetal ovary, and OOSNCR5 had a band in the fetal testis. These results were concerning, and after various rounds of troubleshooting, we decided that there was significant RNA degradation. However, we were able to verify the presence of OOSNCR1 and 5 within the bovine oocytes using the previously isolated proven samples and new RT-PCR reactions. The proven samples produce strong bands. Future studies will focus on retesting the tissue samples and determining if OOSNCR1 and 5 are oocyte specific. Thanks for taking the time to listen to my presentation. If you have any questions, please ask them below.